Okay, I think we'll get started. It's our great pleasure and honor to have David Page visiting us. I've been trying to get David to come for at least 2 years, and he's a very busy man, so I feel very lucky. I have a very long resume to work through, so you'll have to give me a few minutes. No, let me tell you a little bit about David. So his history, he's worked with some really rather impressive people. The late Robert Simpson, he had a stint with as an undergraduate. And then he worked with Ray White when he was in Worcester, who many of you will know is now at UCSF. And also the redoubtable David Botstein. So just looking at that pedigree, you might not be surprised that he turned out okay. His precocious talent was recognized when he became the first fellow at the Whitehead Institute, and he's stayed there ever since, and now has become the director. I think that's kind of a unique feat. It's coming at the bottom and work up. The theme of David's, the sort of overall arching theme of his work is human reproduction. And he's worked in many aspects of this: cell biology, the genetics, the genomics, and has made many, many fundamental discoveries that you'll all be aware of, such as the bizarre and fascinating structure of the Y chromosome, the human Y chromosome, resurrecting the Y chromosome from being this relic that everybody was thinking about, and also some other things you may be less familiar of work involved in working out the basis of sexual differentiation in mammals and assignment of male and female germlines that he's going to talk about a little bit today. I have a kind of a selfish reason for inviting David here. It was based on one of the first times I saw him talk, and he's the only person today I've ever seen being encouraged by the entire audience to go over time in a seminar, at a conference. The only person that didn't encourage him was the guy who was up next. So with that, thank you, David.
Well, thank you for the invitation. It's been, I've had a wonderful day here talking with faculty and with students and postdocs. It's just been delightful altogether. And I think I'm not sure that was an invitation to go over time. I'm not sure that I've got that contract with all of you yet, so I think I'll try not to go there. But what I will say is that the talk I want to give today is a very new talk. It's I've got a sort of a compressed version of some things that I've spoken about before in public, but with any luck, I will get in the last third or so of the first 2/3 of the talk will be preparatory for an entirely new story that I'd like to share with you. And so if it seems that I'm thinking out loud rather than telling you a completely polished story, that's because that's the truth. And I'd also say if there are any really pressing questions as we go along, don't be shy to interrupt. So I'd first like to begin with a little bit of an evolutionary overview, because I tend to think about all these issues of sexual reproduction in a broadly evolutionary context. So first, I should say, how old is sex? Old, right. Well, let's start with easier questions then, like how old is the universe? 13.56 billion. Okay, that's a very precise answer. Thank you. So right, 13, 14 billion. So how old is the planet? 4.5, 4.6. Okay, very good. So if the universe is about 13 or 14 billion years old and the planet is 4 or 5 billion years old, let's, against that, ask how old is sex. So I'm going to suggest that in the evolution of sexual reproduction, there were 4 big innovations or 4 big inventions. The first is the ability to swap genetic information. And I will credit, with taking sexual reproduction to this level, I will credit prokaryotes. We have many ways of swapping genetic information, one of them portrayed here. So I'm going to suggest that sex is on the order of 3 to 4 billion years old. But prokaryotes swap their genetic information in a very free-form way. Actually, I'm beginning to think, is the sound bothering? I think, okay, so let's go to the lights off. Do I? This speaker? That turns on his cell phone. Huh? How about the AV press? Okay, is that better? Okay, right. I thought that the humming ballast was not going to be an issue, but it was starting to get to me too. So I think we'll be happier together. Okay. All right. So it turns out prokaryotes engage in swapping genetic information, but they do it in a fairly free-form way. Sex became institutionalized by eukaryotes, who put genes in pairs and divide them at meiosis. Now, you'd say, why are genes in pairs? Well, one answer might be so we can study Mendelian genetics. But that's probably not the ultimate reason. Genes are in pairs so that you can divide them at meiosis and restore the pairs at fertilization and thereby swap genetic information. And this, of course, means we have an alternation of diploid and haploid phases. And I surely hope that there are at least some students in the lab who can identify this organism. Any guesses? So, of course, we have a yeast tetrad over here and little DAPI images to show these haploid nuclei as opposed to the diploid here. And so I will propose yeast as our poster species for taking sexual reproduction to this point. And I will suggest that meiosis, or genes in pairs, alternation of haploid and diploid phases, I will suggest that that was invented once. It appears to be a feature of the basal eukaryotes. Of course, when you find a eukaryote that does not reproduce sexually, you get to write a paper about it, because it's the exception, not the rule. The rule is reproduction by sexually. And so it looks like this was invented once on the order of 1.5 to 2 billion years ago. So even institutionalized, formalized sexual reproduction is about 1.5 to 2 billion years old. But you know that in yeast, the spores, the haploids, are of two types, A and alpha, and they are structurally indistinguishable. So the next invention that gets piled on top of this is to have dimorphic gametes. And as if one of those gametes is big, we call it the egg. And the little one we call the sperm. And whoever makes the big gamete, we call female. Whoever makes the little gamete, we call male. This is, in fact, the most broadly applicable definition of male and female in the animal kingdom. Do you make the big gamete or do you make the little one? And this was probably, and the point I make is that this invention of dimorphic gametes is piled on top of the earlier, more fundamental invention of meiosis. And I'll suggest that this was invented several times in animals in our own lineage, evolved at least on the order of 600 million years ago. But a more recent invention than the more fundamental one of meiosis itself. But let me put forward now as a poster species for taking sexual reproduction to this level, I will suggest the turtle. Now, why the turtle? Turtle exists as male and female. Male makes sperm, females make eggs, but they don't have the fourth invention, that is sex chromosomes. Turtles, like alligators and crocodiles, have no sex chromosomes, only autosomes. they have temperature-dependent sex determination. Okay? So the point I'm really trying to emphasize is that sex chromosomes are a Johnny-come-lately in the evolution of sexual reproduction. It's an invention that's piled on top of dimorphic gametes, which is in turn piled on top of the earlier invention of meiosis. So of course we can have different kinds of sex chromosome constitutions. And truth be known, I have spent the better part of the last quarter century trying to defend the honor of this little downtrodden Y chromosome over here, but I will not say anything more about it today. In fact, if I mention the word Y chromosome, you should begin to hiss or something like that. Just correct me, because I'm going to try to avoid talking about the sex chromosomes today. But sex chromosomes have evolved independently many times in plants and animals. And in our own lineage, in mammals, our sex chromosomes began to evolve from ordinary autosomes on the order of 200 to 300 million years ago, not long after we parted company with the ancestors of birds. So I'm terribly interested in all of these levels of sexual reproduction. And in our laboratory, we have a lot of studies of the sex chromosomes. But what I want to focus on today is actually levels 2 and 3. I will start by focusing on the question of meiosis, that is the decision of germ cells to embark on the meiotic program in mammals, the question of meiotic initiation. And I'll talk about that in mouse and try to draw some parallels actually between meiotic initiation in mice and meiotic initiation in yeast. And then I'd like to begin to draw connections between this question of meiotic initiation, that is mitosis versus meiosis. I want to connect that to the question of egg or sperm. That is of germ cell sex determination, also in the mouse. So we're going to spend most of our time today up here and then shift to the question of egg sperm. So we'll take the questions of mitosis, meiosis, and then egg sperm. Okay, so if we start with meiosis, meiotic initiation, I'd say how to study meiotic initiation in mammals. Well, I told you already that meiosis looks to have been invented once in the basal eukaryotes. So it's a very fundamental, conserved feature of eukaryotes. So, and of course, I don't need to tell this audience that a really good way to study meiosis in mammals is to start with the orthologs of genes identified by mutational analysis in yeast as playing fundamental roles in meiosis. So though that works well in studying the guts of the choreography of the chromosomes in meiosis, to date, it has not proven a fruitful path for studying meiotic initiation. That is, where does the meiotic program get fitted into the mammalian life cycle? And so, where to begin? About 15 years ago, we began thinking about one possible entree. And it stems from fundamental differences in meiotic initiation, in the timing of meiotic initiation in mammalian males and females. So I'm going to give you a time course here in days of development after fertilization. So from E9.5 out to E10.5, mice have a 20-day gestation. So birth occurs at 20 days. So it turns out the primordial germ cells and the gonadal, the somatic part of the gonad, these two groups of cells, which will eventually be intermixed, they have completely separate embryologic origins. The point I'm making is that the primordial germ cells originate apart from what will become the testis or the ovary. And they actually must migrate to and invade the somatic gonad. And this has been accomplished by about 10.5 days after fertilization. And in fact, at about 10.5 days after fertilization, the table is set for the embryo to figure out whether it's going to be a male or a female. The basic body plan of the mouse is laid down by embryonic day 10.5. The organs are roughed out, but the organism still has not taken a first step towards becoming male or female differentiated. But what happens to these primordial germ cells, which then can be called gonocytes when they arrive as cells of the gonad? What happens to them next? Well, it turns out that if SRY, the sex-determining gene on the chromosome whose name I shall not mention, if SRY is expressed in the somatic cells of the gonad, the germ cells will exit the mitotic cell cycle and arrest in G0. Now, there are a group of students who I had lunch with who are going to remind me that this would be a very good time to explain one thing, which is that the primordial germ cells, in taking this step down the male pathway, do so without consulting their own sex chromosome constitution. I'm going to tell you right up front, germ cell sex determination is not a result of the germ cell's own sex chromosome constitution. Okay? It is a reading of the gonadal soma that they have invaded. In other words, cues from the gonadal soma instruct the germ cells to go down the male or the female pathway. They do not consult their own sex chromosome constitution. Okay, so if SRY is expressed in the soma, the germ cells exit the mitotic cell cycle, arrest in G0. After birth, they resume mitotic divisions, but they don't enter meiosis until puberty. Timing is very different on the female side. On the female side, the germ cells will, by about E14.5, 14 and a half days after fertilization, just a few days after entering the gonad, the germ cells will all initiate the meiotic program. Now, a little bit of a test for you. How far into meiosis? I know we got a lot of students in meiosis here, so this should be very, very easy. How far into meiosis do these embryonic germ cells progress? Diplotin of the first meiotic prophase. Very good. Okay. That's, I was a little disappointed that it wasn't sort of a communal shout on that point, but maybe next time we'll work on this. So, okay. So, and at what, when do oocytes resume their progression through meiosis? At, well, that's the answer to the next question. Okay. At ovulation is the resumption of meiosis, and then it stops again only to be completed at fertilization. Okay? So the point is, meiosis is a long drawn-out affair on the female side, but what I'm really drawing attention to is the difference in the timing of meiotic initiation. So historically, for the last 50 years, what the study of meiotic regulation in mammals has meant has been the study of the mechanisms by which meiosis resumes at ovulation. What I'm talking with you about today is the initiation of the meiotic program, which occurs in embryogenesis in the female and only after puberty in the male. And also, I'll make the point that in the female, at least the prevailing view is that there are no germline stem cells, and all of the germ cells are entering meiotic prophase during embryogenesis, whereas in the male, it's actually the establishment of a spermatogonial stem cell population that cyclically and regularly commits daughter cells to meiosis. Okay, so a very fundamental difference in the timing of meiotic initiation. The question was, could we exploit these differences in the male-female male-female differences in the timing of meiotic initiation to gain molecular entry to the mechanism of meiotic initiation in mammals. Now, I should say right up front, it's by no means obvious that the mechanism of meiotic initiation should be the same in the male and the female. Simply didn't know that going into this, but we thought maybe we could use this difference to gain entry. But if the question is how to gain entry, this idea is nothing more than a hollow shell until it is actually populated by really energetic students and postdocs. And so the heroes of the story are, of this meiotic initiation part, are these four people. So Doug Menke was the first pioneer graduate student. He's now an assistant professor at the University of Georgia. Andy Baltus and Yana Kubova are postdocs, and Erica Anderson will be defending her thesis within the year. So these are four graduate students whose story I want to tell you about in bringing us to where we are today in understanding meiotic initiation. So, I'm just going to highlight, present a few of the highlights of what Doug and Andy and Yana and Erica have learned. So first of all, Doug in particular identified a gene called STRA8 as having a terribly interesting pattern of gene expression. Remember I said we're going to try to exploit the male-female differences in the timing meiotic initiation to try to gain entry. This gene had a really interesting expression pattern, as Doug found. So what I'm showing you here are whole mount in situ hybridizations. The purple is STRA8 mRNA as detected by in situ hybridization. These are dissected testes and ovaries at 1-day intervals across this 4 days of embryogenesis. And what you see with STRA8 is it's expressed in the embryonic ovaries on the right, but not in the embryonic testes. You'll have to take my word for it that this expression is restricted to the germ cells. You'll see that it's expressed in an anterior to posterior wave. It turns out the anterior pole of the gonad is up here, posterior down here. Notice that over the period of these 4 days, expression of STRA8 begins at the anterior pole and over 4 days winds its way down to the posterior pole. It turns out Doug and other laboratories subsequently showed that this expression of STRA8 anticipates by about 24 hours the expression of canonical markers of meiotic prophase like DMC1, for instance. And so it turns out that the germ cells of the embryonic ovary are actually entering meiosis in a wave that sweeps across the gonad. And a really good question would be, what causes that wave? I'll give you the short answer, don't know yet. But have some thoughts about that. So it's a really interesting gene whose expression patterns seem to anticipate meiotic prophase. The question was, is it actually required for germ cells to enter meiosis? So Doug made, in his closing gift to the laboratory's efforts, made a knockout of the mouse STRA8 gene, turned it over to the second graduate student, Andy Baltus. And Andy Baltus and Doug Menke and others went on to show that in ovarian germ cells, that is, in the germ cells of the embryonic ovary, STRA8 is required for meiotic DNA replication, for chromosome condensation, cohesin loading, synaptonemal complex formation, and double-strand break formation and repair. In other words, it's required for the things that define meiosis. It's required for meiotic initiation in the embryonic ovary. It had been shown that STRA8 was also expressed in the spermatogonia, the pre-meiotic cells of the adult testis. And so a question arises, what about meiotic initiation in the adult testis? It didn't have to be the case that the meiotic initiation machinery would be the same in the male and the female, but it turned out to be true, as Erica Anderson showed. So basically in these two studies, this group of 3 graduate students showed that STRA8 is expressed on the brink of meiosis in the female germline, and in its absence, no meiotic initiation occurs, no oocytes are formed. Similarly, STRA8 is required on the brink of meiosis in the male germline, but now in the postnatal adult, and again, in its absence, no meiotic initiation occurs. So STRA8, the whole story that I'm telling you, if there's one central player around which the whole story emerged, it is absolutely straw 8, because it was a mutant with a nice clean phenotype and a very clean expression pattern, and most of the rest of what I'm going to tell you is in one way or another built around the clarity that straw 8 brought to this picture. So what controls straw 8 then? What turns on the straw 8 gene in the embryonic ovary? And here I will quickly summarize the work again of this group of graduate students and some other laboratories as well. Turns out the answer is retinoic acid that's acting through the retinoic acid receptors. Do germ cells in embryonic testes? So remember that straw 8 is expressed in the embryonic ovary, but not the embryonic testis. I've just said that retinoic acid drives expression of straw 8 in the embryonic ovary. Could germ cells in embryonic testes express straw 8 if they were exposed to retinoic acid? Well, it turns out this proves to be true both in gonadal explants. In other words, in this case, Yana Kubova dissected embryonic testes, exposed them in culture conditions to retinoic acid, was able to induce retinoic acid, able to induce straw 8 thereby. And it turns out there are some mutants that Yana has studied that also induce straw 8's expression because they elevate retinoic acid levels. Next question is, are germ cells, if germ cells in embryonic testes can respond to retinoic acid this way, is it that they're not exposed in vivo to retinoic acid? And if so, why not? Well, it turns out that in the embryonic testis, retinoic acid is degraded by a cytochrome P450 enzyme, CYP26B1, that is expressed in the embryonic testis but not in the embryonic ovary. So, right, I'm seeing Ken, you're enjoying this is a fundamental. So if you actually ask from the point of view of what is the job with respect to germ cells either entering or not entering meiosis in the embryonic gonad, what is the key role of SRY? It is to indirectly or directly ensure that this enzyme is activated in the testis. So a fundamental difference between the embryonic testis and the embryonic ovary is the embryonic ovary has lots of retinoic acid, the embryonic testis does not, because the embryonic testis expresses this retinoic acid degrading enzyme. And these conclusions were really captured in these papers. The paper from Bowles et al. Was from the Peter Koopman's laboratory, but these studies sort of captured these conclusions, but they were based on two earlier, terribly important findings. One was that in Pierre Chambon's lab in the mid-1990s, they had identified in the first place the straw 8 gene as being retinoic acid inducible in cultured ES cells. So the straw 8 gene was originally identified in the Chambon lab as one of a group of genes which, when you had ES cells in culture, hit them with retinoic acid, was induced. And so that was the original source of the hypothesis that straw 8 might, in the embryonic ovary, also be retinoic acid inducible. It was based on those in vitro results. And the connection to CYP26B1 stemmed from work of Doug Menke, one of the students I showed you, in combination with work from the Petkovich lab in Kingston, Ontario, which in 1997 had biochemically characterized the CYP26B1 enzyme and had shown it to be a degrader of retinoic acid. What Doug showed then was that enzyme was expressed in the testis, not the ovary, and away to the races. And then what about straw 8's expression in adult testis? It turns out that it's the same pathway, retinoic acid inducing straw 8, in turn inducing the meiotic program. That same pathway is operating, and that was captured in Erica Anderson's work. Okay, so I've sort of quickly summarized a lot of work here. So, and what I want to paint for you is here what I'd call the 2006 model, our 2006 model of how meiosis is initiated in the embryonic ovary. So, of course, the germ cell, its ability to transit meiosis is the defining feature of the germline. So we knew as of 2006 that STRA8 was required for meiotic prophase, more precisely that it's required for meiotic DNA replication upstream of meiotic prophase. And actually, remember, STRA8 is expressed in the germ cells themselves. It's a germ cell intrinsic gene. And we think that retinoic acid, the retinoic acid that is inducing STRA8, is actually produced in the soma. I think the germ cells are responding to a retinoid environment that's established by the soma. And if we ask the question, why does the timing differ in males and females? We actually think there may be several factors in play, but one is that CYP26B1's expression in the embryonic testis degrades retinoic acid, precludes STRA8's expression there. And for reasons, to be honest, we don't understand, the germ cells in the embryonic testis choose instead to exit the mitotic cell cycle and go into a G0 arrest. So that was the 2006 model. And I should say, I may be presenting it as if I no longer think this holds. The model has held up well over the last 4 years, but it had a very serious limitation. First limitation, it left a big question unanswered. And that is, retinoic acid does a lot of things in the developing mammalian embryo. You know any of the other things that retinoic acid is good for? Limb development, lung development, regulating the Hox genes to set up the basic body plan, on and on and on. Okay, so many cell types in the developing mammalian embryo are exposed to retinoic acid. Do they do meiosis? No. Actually, what would be the consequence if they all did meiosis? It'd be pretty ugly. I mean, total body meiosis is not a recommended procedure. You should not try that at home. So the question is, what accounts for the specificity of the germ cell's response to retinoic acid? How to make this very non-specific, potent inducer of differentiation a meiosis inducer. And so what restricts meiotic initiation to the right cells? Here we began to look for clues in yeast, looking for help from yeast. So it turns out that there is an extrinsic inducing signal for meiosis in yeast as well as in mammals. And I think actually this should be a pretty, so what's the extrinsic inducing signal for meiosis in yeast? What do you do to get yeast to do meiosis? You starve them. Okay, so nutrient depletion. So I'm going to suggest that retinoic acid is the analog of nutrient depletion. But you know that in yeast, it isn't enough to be nutrient starved to go into meiosis. First of all, it's a good thing to restrict meiosis to the right sorts of cells. And perhaps the right sort of cell is a cell that's not only starved, but also it's a good thing if it's diploid. And how does a yeast cell know that it's diploid? Well, it asks itself whether it expresses a critical intrinsic competence factor, which is the MAT-A alpha mating type heterodimer. Okay? So so one way to make sure that sort of yeast does meiosis at the right time is to require the combination of the extrinsic and the intrinsic. If we've identified retinoic acid as the extrinsic signal in mammals, maybe there is a counterpart of MAT-A alpha. And I'm going to suggest that it is a gene called dazzle. So let me tell you a little bit about the intellectual history of this gene. So this gene family, the DAZ or dazzle gene family, has been studied in a number of laboratories over the last decade and a half. I'll say that Dazzle is the autosomal homologue of a human Y chromosome gene family called DAZ for deleted anospermia that was identified by Renee Rio-Pera when she was a postdoc with us when she was studying the molecular genetic basis of male infertility, of spermatogenic failure. So this gene family on the Y was deleted in many men who made very few, if any, sperm. The Dazzle gene encodes an RNA binding protein. We don't know with certainty its targets. It's expressed, the Dazzle gene is expressed exclusively in germ cells that have reached the gonad, that have ended their migration. And it's expressed in both male and female, that is both XX and XY germ cells beginning at about embryonic day 11, which is to say very shortly after the germ cells have arrived in the gonad. And it is required for male and female fertility with rather striking genetic background effects. That is to say, there's been some inconsistency in the phenotypes reported on mixed genetic backgrounds, and everything I'm going to tell you about was actually the result of studying a Dazzle knockout on a highly inbred C57 black 6 background. In case you get the idea that that's some odd strain, it's the most commonly used strain in all of mouse genetics. It is the strain that was chosen for the reference sequencing of the mouse genome. So, and what I want to tell you in this story is the work of 3 other, well, actually you've already seen Jana's picture, joined by 2 other grad students, Yanfeng Lim and Mark Gill, and it's really based on the prior work that I've already told you about, including Renee Rio's discovery of the DAZ gene family back in the mid-1990s. And it was possible because of very generous contributions of key reagents, a knockout allele from Howard Cook, and some key antibodies from Krista Hading and Toshiaki Noche. So what I want to tell you is that in Dazzle-deficient embryonic ovaries on a C57 black 6 background, what Yanfeng and Mark noticed was that the chromosomes don't undergo the condensation of meiotic prophase. So here's the wild type at E15.5 wild type ovary. Here are the chromosomes in their characteristic condensation. That simply doesn't occur in the Dazzle mutant. And they were studying the Dazzle phenotype in our laboratory at the same time that another student in the lab, Andy Baltus, was studying STRA8. And this was immediately very reminiscent of the STRA8 phenotype, and then raised the question of, do Dazzle-deficient ovaries express STRA8? And the answer is no. So here's the whole mountain C hybridization at E14.5, wild-type ovary, abundant expression of STRA8, but in the Dazzle mutant, absolutely eliminates, precludes expression of STRA8. So it predicts that Dazzle should recapitulate the STRA8 phenotype. Question, does it? Well, what I'm going to tell you is that it actually recapitulates the STRA8 phenotype, but is even a little bit more severe. And in the last minutes of the seminar, I will tell you that it's actually much more severe. But first, in ovarian germ cells, Dazzle is required for STRA8's expression, and it's required for all these STRA8-dependent events. Basically, it's required for all these events of meiotic prophase. But in addition, Dazzle went one better. It turns out that Dazzle is required for at least one, as I'll show you, one STRA8 independent event. It turns out that the synaptonemal complex protein 3, though it is required, the synaptonemal complex protein plays a critical role in the guts of meiosis. It is actually transcribed and translated well before meiotic prophase. But that depends on Dazzle. So the Dazzle phenotype includes the STRA8 phenotype and shows this additional feature. That is, it is required for SYCP3's expression. And this, if we ask the question, what model of meiotic initiation could explain this, we wanted to account for another observation in the literature. And that is that the De Felici lab had shown in 2000 that even though the germ cells of embryonic testes do not initiate meiosis during embryogenesis, they make SYCP3 protein at the same time that it's made in the germ cells of embryonic ovaries. Now, I just want to let you have this sink in. I mean, SYCP3 has no function embryonically in the testis, but it is expressed in the germ cells of both the embryonic testis and the embryonic ovary. So it's sort of expressed in a unisex way, even though it's going to be used in the embryonic ovary, but not in the embryonic testis. And this is dazzle dependent. Another observation we wanted to make, that we wanted to weave into a model here, is that germ cells in embryonic testes normally do not express STRA8 as judged here. So this is one of Jana's gonadal explant experiments where if she explants a wild-type testis, cultures it for a couple of days, and then assays for STRA8 whole mountain C-tribization, no expression of STRA8, but in the wild-type ovary, like so. But if this experiment is repeated, if Jana repeats this experiment adding retinoic acid, the germ cells of the embryonic testis, of the wild-type embryonic testis, will now express STRA8. Okay? So the point is that the germ cells of the wild-type testis have the capacity to respond to retinoic acid by expressing STRA8. And so a way of weaving all of these observations together was in the following model, which I'll call the 2008 model, which is that the primordial germ cell progresses to a state of competence to initiate meiosis. And from that state will progress in the female into meiotic prophase. That this transition from the primordial germ cell to the meiosis competent state requires dazzle, and that the progression onto meiosis itself requires the extrinsic inducing signal, retinoic acid, which operates on STRA8. Now, and I should say again, SYCP3 protein is expressed, we think, in this phase in both male and female germlines, in both XX and XY, or male and female germ cells, but it's not chromosomally localized. It's only down here that the SYCP3 protein is localized in a way where it does its business. And this made a simple prediction that STRA8's expression in retinoic acid-treated testes should depend on dazzle. Very testable. And so Jana went on to test that experiment. So here is, again, the wild-type testis exposed to retinoic acid in culture expressing STRA8. And the prediction was if dazzle is required, there should be no expression. That's the case. So in the absence of dazzle, the germ cells of the embryonic testis will not respond to retinoic acid by inducing STRA8. So competence, that is the ability to respond to retinoic acid as a meiosis-initiating signal, depends in both sexes on dazzle. So there's the model for 2008. Meiotic initiation requires both an intrinsic competence factor and the extrinsic inducing signal, retinoic acid. Okay, now, so that was sort of where we were in 2008. And the question I'd like to close then with, what are the implications of these findings for germ cell sex determination? So I've been talking about how the germ cells make the decision to initiate the meiotic program. But now there's the question of egg-sperm. So I'm really asking, what is the relationship between the embryonic germ cell's decision to initiate meiosis now or to put it off till puberty? And what's the relationship of that decision to the decision to choose the pathway of oogenesis or spermatogenesis? Or another way of thinking about this is, what do spermatogenesis and oogenesis have in common? They both have meiosis. But an egg and a sperm are very, very different cells. And it appears that they begin to make that decision. They make the decision about which path to follow at about the same time during embryogenesis as they make the decision to either start meiosis or put it off. And the prevailing view has actually been that the prevailing view has been that embryonic initiation of meiosis is the commitment to oogenesis. The prevailing view in the field for 20 years has been that that the mitosis, meiosis, and egg-sperm decision are not two, but one decision. That is, embryonic initiation of meiosis is commitment to oogenesis. So if germ cells lacking dazzle cannot initiate meiosis, one might predict that, well, lacking that ability to initiate meiosis, would germ cells in dazzle-deficient embryonic ovaries begin to show signs of masculinization? Would they begin to follow this path? Okay? And so what I want to show you is the work of Mark Gill and Yanfeng Lin, again, joined by postdoc in the lab Weixiang Yu, to address this question. So first, I need to tell you a little bit about what it means to embark on spermatogenesis during embryogenesis, or to embark on oogenesis. So during embryogenesis, the germ cells of the testis do a bunch of things. First, they express a gene called nanos2, beginning at about embryonic day 13 and a half. And it turns out that nanos2 is both a marker and a driver of masculinization of the embryonic germline. As I mentioned already, they exit the mitotic cell cycle and go into a G0 arrest. So this means, among other things, no DNA replication. And they will not express Ki67, which is a marker of cycling cells. So I'm just going to brace you that Ki67, by its absence, marks cells that have exited the cell cycle. And finally, they remethylate their genome. So here I have to explain that both XX and XY germ cells have demethylated their genome by the time they arrive at the gonad. So they've wiped the slate clean of methylation by the time of arrival in the gonad. And so that's a unisex process. So demethylation is a unisex process. But then XY germ cells begin to remethylate their genome by embryonic day 15 and a half, while XX germ cells wait until after birth. So if you think about it, methylation is kind of the opposite of the meiosis story here. The remethylation. The male germ cells get on with remethylation early. The female germ cells put it off. It's the opposite of meiosis. And so the question then is, do germ cells in dazzle-deficient ovaries display these male characteristics? And just to sort of steal my own thunder, I'm going to give you the answer. They absolutely do not. Let me show you the data. So first, germ cells in dazzle-deficient embryonic ovaries do not express nanos2, which is this male marker and driver. Very simply here, nanos2 in the wild type testis, wild type ovary. This is at embryonic day 13 and a half. Here you see no expression of this male marker in the dazzle mutant ovary. Do they arrest in G0? They don't express nanos 2. Do they arrest in G0? Here, we are looking at BRDU incorporation, the green. MVH is VASA. It is the red marker marks germ cells. Remember, the gonad is a mixture of somatic and germ cells, so it's really helpful to have the MVH mark the germ cells. And what you see is that over here in the wild-type ovary, there are cells that are incorporating BRDU, that are replicating their DNA, but they're somatic cells. In the wild-type ovary, the red cells, the germ cells, are invariably BRDU negative. But in the dazzle-deficient ovary, in the dazzle-deficient ovary, many of the germ cells are incorporating BRDU. So they are continuing to replicate. Instead, they replicate their DNA. They're not acting male. And in addition, I'm now showing you Ki67. So remember I said Ki67 is expressed in cycling cells. So in a wild-type testis, where the cells have exited the mitotic cycle and are arrested in G0, the germ cells are Ki67, which is the green marker here. They're Ki67 negative. And the same is actually true, Ki67 negative in the wild-type ovary, because they're basically arrested in diplotin of meiosis. But in the dazzle-deficient ovary, plenty of Ki67 expressing germ cells. So they are continuing to cycle. They're replicating their DNA. So instead, they express Ki67, a marker of cycling cells. Then do they remethylate their genomes? Final test of whether they're going male. It turns out they do not remethylate their genomes. So here this is using an antibody against 5-methylcytosine. In the wild-type testis, we see, this is at E15.5, we see plenty of germ cells with decent levels of staining for methylcytosine, 5-methylcytosine in their nuclei. This is not occurring in the wild-type ovary, and it is not occurring in the dazzle-deficient ovary. So by all measures, the germ cells of the dazzle-deficient ovary are not male. So in sum, the germ cells in dazzle-deficient embryonic ovaries do not initiate meiosis. They do not initiate male differentiation. So they're neither male, they're not meiotic. Do they initiate female differentiation? Well, it turns out there are very few markers of the embryonic initiation of oogenesis. The best one is a gene called FIGLA, which is a transcription factor, the earliest known factor required for oogenesis. And it turns out germ cells in dazzle-deficient embryonic ovaries do not even express FIGLA. So here's the wild-type testis, which does not express FIGLA. The wild-type ovary does. The dazzle-deficient ovary does not express this transcription factor associated with the beginnings of oogenesis. So what we're left with is that dazzle-deficient embryonic ovarian germ cells do not initiate oogenesis. They don't initiate spermatogenesis. They don't initiate meiosis. So what happens in dazzle-deficient embryonic testes? So basically, nothing is happening in the germ cells of the embryonic ovary in the absence of dazzle. What about in the embryonic testis? Well, it turns out that germ cells in dazzle-deficient embryonic testes don't express nanos 2. Here's the dazzle mutant testis. Do they arrest in G0? No. Here are the germ cells of the dazzle-deficient testis still showing BRDU incorporation. They should be stopped. But in the absence of dazzle, they're still incorporating BRDU. Oh, sorry, this is Ki67, excuse me. They're expressing Ki67, the marker of the cycling cells. Do they remethylate their genome? They do not remethylate. So again, here's the wild-type testis showing staining. and in the dazzle-deficient testis, not showing the staining for 5-methylcytosine. So there is no germ cell differentiation in dazzle-deficient testes or ovaries. No spermatogenesis, no oogenesis, no meiosis. So how to make sense of this? I'd like to propose a new model of embryonic germ cell differentiation based on these findings. So I'll suggest that in germ cell, in the embryonic development of germ cells, there are two big processes that are generally accepted. One is the process of germ cell specification, which is occurring around embryonic day 7. Cells of the proximal epiblast give rise to primordial germ cells. Those germ cells then migrate to the gonad. I've just talked about that already. What I'd like to suggest is that there is an additional process not previously appreciated that I will call here licensing for gametogenesis, a process that is mediated, that requires dazzle. And that the result of that licensing process is a cell that is now competent not only for meiosis, but that is competent for both the processes of oogenesis and spermatogenesis that together define gametogenesis in the two sexes. So basically what I'm suggesting is that germ cells that are lacking dazzle are specified, migrate to the gonad, but once they arrive in the gonad, they don't have a clue why they're there. They have no idea as to how to embark on either spermatogenesis or oogenesis in the absence of dazzle's function. This makes one nice big prediction. It predicts that primordial germ cells lacking dazzle will migrate but retain the characteristics of primordial germ cells. That's very testable because there are plenty of gene expression markers of primordial germ cells. It turns out that primordial germ cells express a whole suite of genes that are more typically associated with pluripotency, genes that are typical of embryonic stem cells. And so I'll just show you here quantitative RT-PCR data that Mark Gill generated, time course from embryonic day 12.5 to embryonic day 15.5. In this case, the blue curves are wild type ovaries. The red curves are dazzle-deficient ovaries. What you see is actually the migrating PGCs which are proliferating at this point, they express high levels of these markers that are characteristic of and you may know better as markers of pluripotency associated with ES cells. OCT4, NANOG, SOX2, and this DPPA3 is also known as Stella for the germ cell aficionados in the audience. And what you see is that all of these genes in the wild type decline, their expression levels decline dramatically upon reaching the gonad and a few days after. But in the dazzle mutant, those expression levels plateau, stay high, they simply don't go down. So in other words, these dazzle deficient germ cells are retaining the gene expression characteristics of primordial germ cells. And just to show you that this isn't based just on quantitative RT-PCR, here is the whole mount in situ hybridization for OCT4 in the dazzle mutant compared to the wild type ovary. This is at E15.5. And then to switch over to the testis, looking at E15.5 for the, by this point, the expression of OCT4, this is immunohistochemistry, the expression of the OCT4 protein is diminished dramatically by E15.5, but remains robust at E15.5 in the dazzle mutant. So I then would like to just close by coming back to this model. So again, we think that there is a that Dazzle, which as of 2008, we thought was required to make the cells, the germ cells, competent to respond to retinoic acid as a meiosis initiating signal. We still think that is the case. It absolutely is the case that germ cells lacking Dazzle cannot express striate in response to retinoic acid, cannot initiate meiosis. But what I'm telling you is that that's not just limited to meiosis. It is, in fact, sort of a gatekeeper to the whole of the downstream processes of differentiation, both spermatogenesis and oogenesis. And with that, I will stop, and I'd be delighted to take your questions. Thank you. Any questions? Yeah.
So it seems like there has to be some other factor involved here too, because in between the PGC and overt initiation of meiosis, you have this transient stage where cells divide with incomplete cytokinesis. So it sounds like Dazzle mutants are blocked prior to that that stage where cells continue mitotic division with incomplete cytokinesis prior to overt differentiation. So there needs to be another step or another factor that regulates when, after you initiate down this transient stage, when do you stop to actually overtly enter meiosis?
Well,
does that make sense?
But let's do it again. So you're talking about the incomplete cytokinesis.
Yeah.
So how does the incomplete where would you like to?
So Dazzle mutants are blocked at the PGC stage. So I'm assuming that if you looked at their germ cells, they would not be present in cyst.
I'm not sure of that actually. I'm not sure of that actually. But so you're saying that should occur in. Well, is that occurring in both sexes?
Yes.
Well, so what I would suggest is that Dazzle, we don't see any evidence that the unisex processes are failing to occur in Dazzle mutant. What we're seeing is that the process that actually involves sexual differentiation are not occurring in the Dazzle mutant. So remember, one of the things that's occurring during migration is the demethylation of the genome. That's a unisex process which looks like it's more or less being completed as they, right. Yeah.
You would, in the embryonic ovary, you would see this transient stage of incomplete cytokinesis.
Mm-hmm.
In males, you wouldn't. They would arrest at single cells. You would only see that stage after puberty.
Mm-hmm.
So that's
but the question of how to connect that, maybe we should take this up afterwards. I'm not exactly. So you're saying, well, first of all, I'd say there may well be many more additional steps in here. I'm not in any way philosophically opposed to further subdivision of the process. What I'm really getting at, and I thank you all for staying with me. This is the first time I've presented this particular information. What had been thought was that once specified, the migrating primordial germ cells are essentially set up for oogenesis or spermatogenesis, right? And there is some old literature that suggests that germ cells that mismigrate, that don't even make it to the gonad, can embark on oogenesis. But of course, there weren't really very many markers used in these studies that were done a few decades ago. So what I'm really suggesting in a way is that this specified primordial germ cell it knows to migrate to the gonad, and along the way it's going to demethylate. But it really doesn't have a clue as to what it's going to do when it gets to the gonad until Dazzle is expressed. That's the idea I'm entertaining. Yeah. Yeah.
What is the evolutionary breadth of Dazzle? And maybe you don't know the function.
So Dazzle. So it turns out that genes of the Daz family have been found in all metazoa.
But is Dazzle a separate sort of sub-branch within that?
It's no. I mean, it's a pretty tight. So this is these are members of the broad realm of RBM. RNA binding proteins, but they are a very discrete, tight little family. And not only are they separable in sequence space, but basically the genes, to my knowledge, the genes of the DAS family throughout metazoa, whenever their expression patterns have been studied, they are found to be germ cell specific. So it's a very germ cell specific, there's an amazing germ cell specificity of the expression pattern of the DAS gene family throughout metazoa. In addition, where loss of function mutations have been generated, and they have been generated in a host of metazoa, the result is invariably infertility in male, female, or both species, or both sexes. But the precise functions of DAS, that is the nature of the phenotype, differs among the species. But there's a high degree of conservation in metazoa of the germ cell specificity and the requirement for fertility. Yeah.
Whereabouts along this scheme are the stem cells of the testis?
Okay. So what a whole.
They remain not in meiosis, but able to go into meiosis.
Right. So basically, the stem cells of the testis are lurking under this word spermatogenesis. Okay. So basically so I've just been showing you pictures of embryonic testes and processes that are or not occurring in the germ cells there. By basically the spermatogonial stem cell population of the testis is established in the mouse during the first week or two after birth. They find their niches in the first week or two after birth. And then those cells will, in a very, very regular way, commit daughters to the spermatogenesis process, which includes meiosis, throughout a male's adult reproductive lifespan. I have a feeling I'm not explaining. I mean, the intricacies of the. So it turns out that STRA8 is expressed in the postnatal male germ cells at a couple of points. And we're actually entertaining the possibility now that STRA8 may have multiple functions. It may function at multiple points postnatally. The strongest arrest that we see is actually at the point of meiotic initiation.
So this huge difference between males and females, is that part of the explanation as to why there are not stem cells in the ovary?
You mean, does part of it come down to STRA8 and retinoic acid?
That you make the commitment to meiosis early and you don't save any cells that could be stem cells, basically.
I think that is part of the difference. It would be convenient if there were a simple, a sort of single gene switch between establishing the stem cells or not. I suspect that evolution has had opportunity to sort of reinforce these mechanisms in multiple ways. So, for instance, just to give you an example, nanos-2, I mentioned nanos-2. It's expressed in the germ cells of the embryonic testis. It is thought that one of the functions of nanos-2 is actually to translationally suppress STRA8. There may be multiple mechanisms that are reinforcing the decision to not initiate meiosis in the male during embryogenesis. So it's a great question, but I suspect that there's not a singular mechanism. There's not a single mechanism which, if you knocked it out, you would suddenly find yourself with ovarian stem cells, or sort of vice versa. But the question of how the spermatogonial stem cells of the adult testis are regulated is in and of itself a really, really rich and wonderful question. It looks like STRA8 and retinoic acid are playing key roles there as well, but I haven't even touched on that. But it's all of that is lurking under this one word and one arrow out there. I need to flesh that out more fully for you. Yeah?
So you mentioned that retinoic acid is degraded by cytochrome P450. Is that a redox reaction?
It's actually that's right. So it doesn't actually it actually leads to essentially the cells getting rid of the retinoic acid. It's not actually so much degrading. I've sort of simplified there, but I'd be happy to go over those reactions with you if you'd like. But it essentially results in effectively inactivating the retinoic acid. And there's a collection. There are actually 3 different enzymes of this CYP26 family. It turns out that CYP26B1 is the only one that is expressed in the embryonic gonads. Now, you may be thinking about, so it turns out in the adult testis, where there's a very tightly regulated cycle of spermatogenesis, evidence is emerging that there may be a spatially and temporally regulated, tight spatial and temporal regulation of retinoic acid levels. Okay, so the testis there might be a good bit of retinoic acid cycling going on in the adult testis. Yeah?
I apologize for the question because it's not directly related to your talk, but you're the best person to ask this.
Does it have to do with politics or?
Healthcare. Well, we could get there if you wanted to.
Okay, well, I'm just worried about what I might be the best person to answer.
There's some old data in yeast that suggests that meiosis as a process is mutagenic. Mm-hmm. And there are some newer data that could be constructed to reflect that as well. Now, it's very difficult to estimate mutation rates in mammals because there are so many generations involved in the generation of the germ cells. And so I was wondering, from your point of view, what's the best estimate in humans and mice of how many generations you have that lead to the generation of the male or mouse sperm or so?
Yeah. So the question of is the question how do the male and the female germlines differ in this respect?
Yeah. Also, just the sheer number of how many cell generations.
Yeah. Well, okay. So the trick is so recognize that in the female germline, entering meiosis during embryogenesis, and in the male germline, establishing a spermatogonial stem cell population that's mitotically dividing, there are in the male germline of mammals far more cell generations on the male side than on the female side. And if I remember correctly, it's I'm going to say it's something like, on the female side, it may be something like, don't hold me to these, I'm just trying to, I think on the female side there may be something like 20 mitotic divisions in a life cycle on the germline. And in the male, it's upwards of, it's a minimum of, I'm thinking 35 or 40. And can go much higher because it's a function of how old is the male. And so this has led to, there are a number of diseases in human genetics where the incidence is a function of the father's age. Okay, so we tend to be obsessed with the role of mother's age in some meiotic non-disjunctions, but there actually are a number of disorders that are autosomal dominants that involve base pair substitutions, where there is a striking correlation with paternal age, and with that, presumably with the number of mitotic divisions in the male germline. Although, to be perfectly honest, the curve rises exponentially with father's age in a way that's not easily explained by a simple linear increase in the number of mitotic divisions. That's a great question, actually. Yes?
So you mentioned that early on at the decision to go either the oogenesis or the spermatogenesis, there is this wave in the striate that starts at the posterior.
At the anterior, and over 4 days sweeps posteriorly. Yeah.
So is there a degradation factor or something intrinsic or extrinsic, you would think, that causes that wave? And could that be also involved in the decision at that point?
Well, so it's a great question. So the question is, what might account for that anterior to posterior wave of STRA8 expression and of meiotic initiation in the germ cells of the embryonic ovary? I think of the possible explanations in two groups. One is, the one that occurred to us first is, well, if retinoic acid is the meiosis initiating signal and it's driving STRA8's expression, maybe there's a wave of retinoic acid that sweeps across. So one would be, maybe it's the inducing signal that sort of flows across the ovary in a wave. Another possibility is perhaps there's a wave of competence to respond to retinoic acid. So, questions about, well, maybe the germ cells in the anterior were the first to reach the gonad, and they're a little bit more mature than the germ cells that reach the posterior end of the gonad. So maybe they are a little more ready to receive that retinoic acid signal at the anterior pole. So maybe it's an inducing factor, maybe it's competence, or maybe it's some combination, or maybe it's something of another category of the sort that you suggest. But we fundamentally do not understand the basis of the wave at this point.
Does it have any function or significance with that?
Well, we haven't figured out how to eliminate the wave. So we don't yet know, as somebody who's obsessed with gaining insights through mutants, we don't yet have a mutant that eliminates the wave.
It's also seen later on in the testes.
Oh, is the wave seen in the testis? So, well, that, now you get me really going. I'll try to keep the answer short. So again, the spermatogenesis in the adult testis is a very, very regularly cyclical process. And it involves waves in time and space. There are definitely waves in time and space in spermatogenesis. And work from a number of laboratories, including our own, is beginning to hint at the possibility of actually an oscillation of retinoic acid levels is playing a role in that process. So, but now whether we want to connect the wave in the embryonic ovary to the waves in the adult testis, there may well be a connection, but since we don't understand either of those waves very well as yet, it's just speculation that there may be a connection. Yes?
Can you knock out the enzyme that degrades the retinoic acid?
Yeah, CYP26B1, we knock it out.
Get the male gonad-producing oocytes?
Ah, great question. So, right, so if you, so I said that that CYP26B1 is not expressed in the embryonic ovary. It's expressed in the embryonic testis, degrades retinoic acid, precludes STRA8's expression. So if you knock out CYP26B1, and I showed you an experiment that Jana did with gonadal explants, where she exposed the germ cells of the embryonic testis to retinoic acid in vitro. The CYP26B1 is the in vivo moral equivalent of that experiment. So first of all, what should happen to STRA8 in the CYP26B1 mutant testis? Should go up. It does go up. So first, that's really nice to know that in vivo, you recapitulate what you see with the gonadal explants. Now, the question is, those germ cells in the embryonic testis, now essentially retinoic acid poisoned, if you will, by knocking out CYP26B1, what do those germ cells do? So now we sort of think about what do we predict them to do? Well, maybe they're going to initiate meiosis. And it turns out It turns out it looks like they put their toes in the water of meiosis, but don't get very far in. I think that there are other blocks to full-blown meiotic prophase in the germ cells of the embryonic testis. And then beyond that is the question of, do they express Figla, that transcription factor associated with oogenesis? They do not. Okay? So what I'm suggesting here is that this gametogenesis competent cell is set up to now receive signals from the somatic gonad, be it a testis or an ovary, that will drive those cells down the oogenesis or the spermatogenesis pathway. In addition, on this, and one of the components of this pathway is meiotic initiation. Other questions that I have not addressed here today lie ahead, and I hope that the mutants arise to answer them. One of them is, so in a sense, I started out with this sort of prediction of the prevailing dominant model. That is that if you can't do meiosis, that you should go down the male pathway. But that prediction was premised on the prevailing view's notion, on the prevailing view today that embryonic initiation of meiosis is a commitment to oogenesis. Maybe there will be mutants that will allow those two to be separated, or at least allow those two, the connectedness of those two processes to be tested. And so I think we've got any number of processes that are playing out in time in parallel. And there have been, in the absence of mutants, over the last several decades, the assumption has arisen that these are all regulated in exactly the same way. But if you ask hard, what is the evidence to support that assumption? It does not exist. It's simply the descriptive biology that in the absence of mechanistic insight leads to a simplifying assumption of a single regulatory scheme. But I personally think the single regulatory scheme is unlikely, especially based on what emerges from genetic studies in model organisms. Yep.
Any role for sex steroids in any of this early embryonic specification differentiation?
Well, you know so let's think about what are the sex steroids? Estrogens. Yeah, yeah. Well, let's just stop for a moment though and think about this. So, estrogens, androgens, how do they function? What are estrogens and androgens?
Well, they're like retinoic acid, isn't it?
They're ligands for nuclear hormone receptors. So first of all, I would say retinoic acid is another sex hormone. And I actually find it an interesting parallel in a way to say that in thinking about sex differentiation, we've got to think about three broad classes, at least three broad classes of ligands for nuclear hormone receptors. The traditional ones, the estrogens, the androgens, but now I'll add retinoic acid to the list. And it's interesting to think about the different chemical properties of these substances and how retinoic acid might be better suited to some of these tasks. So for instance, retinoic acid, by contrast with testosterone, with androgens and estrogens, which are both capable of working at a considerable distance in the body, retinoic acid is a local actor. So if you're thinking about controlling this cycle of spermatogenesis, for instance, tightly controlling it in time and space, you couldn't possibly do that with estrogens or androgens. But retinoic acid might be up to that task. But back to your question more directly, where do androgens fit into this scheme? Well, it looks like, believe it or not, we don't, but maybe you know more about this subject than I do, but my reading of literature suggests that, indicates to me that the role of androgens, even later in spermatogenesis, is a bit of a black box. You can clearly mess up spermatogenesis with levels of testosterone that are too high. But we don't see any, I am not aware of any evidence to implicate either androgens or estrogens in these processes that I'm laying out right here. They have profound effects on somatic sexual differentiation. Actually, I'm trying to remember. I think the germ cells are actually pretty free of receptors for androgens. So they seem to have stayed out of that game, left those sex hormones to the somatic tissues.
Okay, drinks and dinner are awaiting us.
Okay,
Just like
thank
to
you all.
David again. Thank you.